Hello, I'm now going to be walking through how to do a general linear model using NIRS Kit. Going to go ahead and share my screen. All right, so in my opinion, the general linear model function is one of the best parts about working with NIRS Kit. It's very useful to have that built-in feature and not have to do it yourself. So I highly recommend that if this is your first exploration of your data set, the first kind of post-processing technique you use is the general linear model. Um, on a simple level, what the general linear model is doing is taking a look at your fNIRS data and comparing it across different conditions, and then you can use your results to make conclusions about whether or not certain conditions led to significant changes in brain activity in each channel. So the first thing that you need to do before jumping into running the GLM, as I'll refer to it, is you first need to uh, make what's called a design inference file. So this is something that's discussed more in depth in the NIRS Kit user manual. So that is a great resource to refer to if you're making your design inference file. But basically what it should look like is you should have a column with your participant IDs, and then you should have at least one column, maybe a few columns for however many conditions that you measured. For me personally, this is kind of a unique data set to run a GLM on. Usually, GLMs are done with a block design, which means that you probably have multiple baseline periods and then multiple trials of the same condition. So you would alternate between a rest period and then a trial and then a rest and then a trial. And then the GLM will look at the effects of each trial altogether. For me, my data set is a little bit different. I have only one rest period and one trial. So regardless of how many different types of events you have or how many different trials of each event that you have, you're still going to set up your design inference in the same way. So for each of your conditions, they'll have a unique column. The first value that you see in this cell is going to be the start index of that event. Um, so for this particular person, their task started at the 12,000th index approximately. And then the duration of that event is going to go into the second cell. So this person did this event for approximately 9,000 samples. And then you can see we've done that for each participant. And you may be asking yourself right now, well, I thought you mentioned you also had a rest period. Why aren't we seeing that here? For our GLM, we consider the rest a baseline and not a condition. So that's why you only see one column here. However, you can design your GLM to be reflective of however you want to consider each of your conditions. So for us, we just have the one condition, and that's what we're going to be looking at. Now, if you're wondering how I generated this file from our SPM files, there's a couple different ways to do that. What I personally do is I write a MATLAB script to kind of do all this math for me. Um, so basically what the design inference script that I've written does is it looks at the total length of our data file. And then for each participant, we use the multiple conditions file to extract onsets for each event. And then we can kind of use the length of the data file and then the onset of the event to conclude how long our event lasted. Um, If you have multiple events, what you probably will do is, use each start index to do some logical indexing and put together this picture of when your events start and how long they last. You can get creative to do this. There's, like I mentioned, more documentation in the NIRS Kit user manual. But yeah, basically what you need to do before you start the GLM is create your own design inference file that matches this format. And then the other thing that's really important to note is that, of course, as I mentioned in my previous video, you have to always prepare your data before using it in NIRS Kit. So maybe you didn't do any data pre-processing in NIRS Kit, but you certainly need to prepare the data before you open it for the GLM. And then your data should also be named in the same way that the names are shown here. So it's important to only have your four-letter participant IDs as your file names. And I mean, as opposed to maybe, how your files were exported, they may have these other appendices at the end of the record ID, but what you need to do is manually rename the files to make sure that the file names are exactly matching what's shown in your design inference file. So once you have those two things set up, you can go to this individual level analysis tab in NIRSKit. You're going to add your data path, which for me is pre-processed data that I had applied motion correction to, and then also a zero point 0.1 hertz low pass filter. If you're curious about that, you can watch my previous video. Okay. So here you can see all my subjects. You also have the option to add covariates. My understanding is that this is mostly used to do short channel correction. I haven't explored that feature yet, but it certainly is an option that you can read more about in the user manual. You're going to want an output folder to save the results of the GLM. So I create this folder called GLM output, and it will create these subfolders for you. So all you need to do is create some sort of GLM output folder. I personally only look at oxygenated hemoglobin, so I'm going to go ahead and uncheck this deoxygenated hemoglobin option, but that's up to you and your preference, which fNIRS outcomes you're choosing to study. It only gives you one option for the hemodynamic response function. The preset here is a canonical hemodynamic response function, which to my understanding is extremely conventional with fNIRS data. So you don't probably need to worry about that. This is important here. So for me, my design inference varies from subject to subject. And what that means is that the start index and the duration of each condition is going to be slightly variable from subject to subject, as you saw in my design inference folder. So I'm going to click different here. And then I'm going to load my design inference. It seems like I just got an error. Let's see what that's about. Okay, I had a little error in my data folders. I just resolved that. So let's keep moving forward with this. Okay, so now I successfully loaded my design inference folder. I just selected that, and then it set up this little contrast for us here. So basically what what this is saying is that we have in our data recordings, the start of our file is this, white area. For my data, I start with the rest, so this makes sense. And then our condition flips on in the second half of the data recording. So this is just visualizing that for you. You're going to want to set an output folder. Like I mentioned, that should be your GLM output folder. And then the last thing that you're going to need to do is define the contrast. So you can name this anything. I'll name mine, dual task because it's the only condition that I have, but you could literally name it anything. Sometimes I just am lazy and I'll just letter it A because it doesn't really matter what it's named. And then what I'll do is I'm just going to tell NIRS Kit my hypothesis. So I have one condition and I have one rest period. So what I believe will happen is that there's going to be an increase in activation during my task, put a one, and then my rest is my baseline, so I expect like no change in activity there. Um, you have other options here, so the important things to note, first of all, is that you'll always define the contrast for the conditions first, and then the last value that you type should be for your baseline. That's why I put 1, 0 as opposed to 0, 1. And then, it might be the case even that you have some sort of situation where you expect to actually see deactivation due to your condition. So you would put -1, 0. If you have multiple events, you can type multiple things here. But your last value should always be a 0 because you have some sort of baseline that you're comparing things to, and that would be the one instance that, there's not going to be any change to your brain activation. So like I mentioned, for me, I expect, my task to increase their brain activity, and then all I have is my baseline period. So this is how I always set up my contrast. And then once you have all of this ready, you're going to press run. I'm not going to run mine right now for the sake of time, but I can show you now what these outputs look like. So when you run the GLM output, you'll get these two folders, GLM and Oxy. If you selected deoxy or total hemoglobin, you'll see those folders here too. I only selected oxygenated hemoglobin, so that's what I'm gonna work with. I can go into this folder. I'm mostly concerned with this folder. Um, my primary focus of my analysis is to understand how the dual task is affecting changes in activation. So what you're looking at now is going to be the beta values for each channel. Let's open one of these up and take a look at an example. Index data is what it should be called. And then my indices. So beta values are kind of a the simplest way to think about it is when you're running a GLM, what you're doing is sort of fitting a regression to your fNIRS signals. So if we're thinking in very simple terms, we can think of, the classic linear regression formula, y equals mx plus b, the slope of a line. Um, we can use that as an analogy for our beta values. So the y in that formula pertains to our fNIRS signal, so we're trying to understand what sort of factors influence our fNIRS signal. And then your x is going to be your different channels, And then each channel is going to have its own coefficient of activation. In the example of y equals mx plus b, you can think of m as your task beta values. And then, of course, there's also going to be a constant, which is the b in that formula, and that pertains to these beta naught values. People don't commonly necessarily investigate or report those, but you certainly can take a look at them. So what I'm looking at here is the beta values for one subject. I have a 22 channel fNIRS system, so I have 22 different beta values, and you can see there's a big range here in our values. So if a beta value is close to 0, what that means is that there wasn't really any change in activation there. If a beta value is greater than 0 and probably even closer to 1, that's usually indicative that that channel was seeing an increase in activation. You also will sometimes see negative beta values. Obviously, this one is very close to 0, but if you saw -1, for instance, that would be more indicative of deactivation in that brain region for that channel. So usually what you'll want to do, depending on what kind of analysis you're doing with the data, but for the most part, you're going to want to do kind of like a group level investigation of these beta values. So within my own data set, I have a couple of different groups that I've defined to stratify my participants. So what I might do, for instance, is get the average beta values for each channel across my groups. You also have some ways to explore these results in NIRS Kit. So there's this group level statistics button. You can see here it allows you to do some statistical testing. So if you're going to do a one sample t-test for your beta values, basically what you're going to do is, the null hypothesis would be that all of your beta values are 0, and then you can do a statistical analysis to see if there are any values that are statistically significantly different from 0 for your groups. That's one way to do it. You can also compare your beta values across groups using the two sample t-test. There's also a way to do a one-way ANOVA to compare multiple groups and add covariates. Of course, you can do all of these statistical analyses in external softwares too. I know lots of people like to use R or JMP for their statistics, especially if you have a more complex statistical analysis in mind. It might be better to kind of get out of NIRS Kit now. But what you can do if you want to do any sort of analysis in NIRS Kit is use this beta 1 folder. That's where all of your indices are stored. What you might have to do is create multiple folders if you want to look at differences across groups. I may have done that at one point here. Yeah, so I have a GLM output by group. I put some subjects from one group in here, some subjects in a different folder, and that way I can kind of do like within group testing and then also between group testing. And these are the folders that I would add as my individual indices. I would select one of those folders, and then select somewhere to save it. And then, like I mentioned, you should set your base to 0 for the beta values. And then there is one more thing that I really like to use NIRS Kit to do once I've done my GLM. Like I mentioned, I usually do between group comparisons. So here I would calculate the average for each of my groups using this function. I'd put in a folder for my individual indices, put a save path, and then what it will do when you click analysis is it will save you this full file. I name mine average. I can open it up, and then what you'll see here is this stat data struct, and it will have the results of the average of whatever participants I had put in that group. So once I have that average calculated, NIRS Kit has this really awesome feature that allows you to visualize your results. So I'm going to go to results visualization. I'm going to input my average MATLAB file that I just opened up and showed you guys. It's going to auto set the range for me based on what the minimum beta value was and the maximum beta value was. I tend to prefer to change this usually. I don't know why that just happened. Okay, I usually prefer to set mine to something more like this, because usually I'm comparing across groups, so I don't really want to use different scales for my groups. But depending on what you want to do with the figure that it's about to create for you, you can decide yourself what the appropriate range of beta values is. So here's for one of our groups. This happened to be like our control group, so it makes a lot of sense that we see lots of activation during our test. So you can see here that, the maximum value I set it to was 2. So these darker red colors are going to show there's increased activation there. Once again, these people were doing a task. So I would not expect to see any deactivation. So that makes a lot of sense that I don't see any blue colors. Now, on the other hand, this data set that I'm working with, we also have a group of people who have some cognitive impairment. So just to give you a frame of reference, if we wanted to look at our other group, we'll set the same range, -2 to 2. Let's see what this looks like. Okay, wow. So this also makes a lot of sense. We have, you know, a group that's not cognitively healthy. They're actually showing some task evoked deactivation. Um, these figures are great to include if you're planning to publish any of your results. They're very nice. You can also, map, you know, change the colors if you want, depending on how you like it to look. I like the blue and the red because it's intuitive to me that red means like increased activity, blue means decreased activity, but of course that's totally your personal preference. And it's also really nice to do this just to kind of get a read of what's actually happening with your data. It's just a quick visual check, like, okay, these results make sense, these results don't make sense. And yeah, that's pretty much what you can do with the GLM in NIRS Kit.